Welcome to my poster booth. My name is Gidon Mamo, and I'm going to tell you about the organization of outer membrane protein biogenesis. First, let's look at the outer membrane. The outer membrane is rigid and lacks energy sources, yet the cell has to insert hundreds of thousands of protein into the outer membrane every time it divides. All this hard work is being done by the BAM complex. This complex is essential for bacterial viability and recently became a common target for new antibiotics. Using unique labels, we were able to visualize BAM on the surface of live E. coli cells. We found that BAM forms clusters on the surface of the cell, and the clusters contain around 10 complexes. These clusters are immobile and uniformly distributed across the outer membrane. But surprisingly, when we looked at OMP insertion sites, we noticed that they are not uniformly distributed at all. In fact, OMP biogenesis is mid-cell biased and cell cycle dependent. And when we compare the sites of OMP insertion to the localization of the BAM machinery, we see that they are very different. So how can we explain this disparity? We looked at several processes in the bacterial envelope and found that OMP biogenesis actually mirrors the insertion of new peptidoglycan into the cell wall. Our latest collaboration displayed that BAM itself directly binds to the cell wall, and this binding modulates its activity and therefore shapes the patterns of OMP biogenesis. So to summarize, we saw that BAM distribution considerably differs from that of OMP biogenesis. OMP biogenesis actually mirrors the process of peptidoglycan biogenesis into the cell wall. And the similarity between these two processes results from the fact that BAM binds to the cell wall and changes its activity rate. Thank you for coming to my poster booth. Looking forward to seeing you in the meeting.